characteristics of a bacterial DNA molecule. It can be supercoiled and circular. They have a single chromosomal copy. We talked about the anti-parallel complementary strands, and anti-parallel, as we've already talked about, is in relation to the chemical ends being opposite to each other. The other thing is down at the bottom, everybody should know these 4 different bases that are associated with DNA. Right. We also talked about how these are grouped into 2 different chemical classifications as far as the bases are concerned. That was called the purine nucleotide and purine thymine guanine. The pyrimidines are cytosine and thymine. The part that's really important to remember is the base pairing. Okay, so the base pairing rules associated with DNA strands include A is always going to pair with T, G is always going to pair with what? C, right? Make sure you remember how many hydrogen bonds exist between each of these pairs. So you got 2 for A to T, 3 for C to G, and so this means, again, the more bonds you have, the stronger it is. Okay. What you will see on the test is going to have something like this. It's going to have a strand, and you're going to have to base pair it, okay, with another strand that's provided for you in multiple choice selections. Okay, so just make sure you remember the base pairing rules associated between each of the bases that we just highlighted. I'm not going to have anything like this on the test for you guys to label or anything, but it does show you the relationship between the 2 strands in this illustration with having the anti-parallel ends. We have the 3' end and the 5' end for each of the strands, both left and right, that are facing opposite to each other. And then it also shows you the interaction between the bases that are on opposite strands, forming the hydrogen bonds between the 4 bases that we just talked about. So the other part of this is to always remember the orientation associated with 3' is always going to face opposite to the 5' end of the opposite strand. So if you have, this strand written out like this, okay, as you see at the very top, and let's just say if there's another strand that is written like this, is that the same as that second strand? So the first strand is on the top. Is this the same as the second strand that's directly below it? Why not? Is it the same or not? How many say no?
It's the same. It's the same. Just run backwards.
It's the same. It's the same. Because it's in reverse. Right? So where do you always start at?
35.
That has to match with what end of the provided strand. Has to match with the 5' end, right? So if you go to the 5' end and you look and you say, okay, that's A, T, G, C. Take a look backwards. Does everybody understand that? So it's not going to be given to you, like you would see it like this. It's not going to be like this. Because you will have the strand facing in this direction. That's going to have the 5' ends never match up, right? You never put the 5' end of one strand adjacent to the 5' end of the other strand, right? Because they have to be facing opposite to each other, okay? So that's something to think about, all right? So, rest of this, don't worry about. Now, as far as some of the information on this, the bidirectionality, remember that this is associated with the circular molecule of DNA and bacteria, makes it much faster. So you have 2 different replication forks. And then semi-conservative, remember what that means, is that each of the daughter cells are going to get one of the original strands and then what? A copy of that strand, right? Down at the very bottom, so this was the part that everybody should remember because we've been going over and over and over this stuff. in relation to elongation of the new strand is going to occur in which orientation? 5 to 3. And the template, or the original strand that's being copied is read 3 to 5. And it's read 3 to 5, right? We, looked at this, origin of replication. Remember that that's the site where DNA replication is initiated for all DNA molecules. This shows you the bidirectional two forks that are splitting down each opposite ends of the strand. And then at the very end, we get, of course, the two daughter cells that are inheriting one of the original strands, and a copy of that strand services the semi-conservative aspect of DNA inheritance into the daughter cells. So, you think there any questions on this? Yes, of course. This is part of your homework, so you need to make sure you know those 5 enzymes, what they're involved in. As far as their function associated with DNA replication. You should also remember that there are the Okazaki fragments related to being on the lagging strand of the replication fork. And then the leading strand is always going to be continuous, all right? There's no breaks in it. Okay? And I think that's it. I don't think I'm going to have anything for you guys to match up label-wise or anything like that. Just make sure you know the definitions associated, the functional roles associated with those enzymes we talked about with DNA replication. Any questions? Continuous strand that's being copied, our name is on the leading side of the replication fork. Lagging side is going to have the discontinuous side resulting in Okazaki fragments.
Could you go back to the slide that has the bidirectional?
That? Okay. All right, any other questions? All right, so moving on. We have the second part of that, which was looking at genetic expression. Okay, so this was looking at genetic expression into proteins associated with the characteristics of the cell. We talked about the 2 steps that are associated with producing a protein in the cell, coming from the gene that's housed in the DNA, going through transcription, which involves RNA polymerase, generating the messenger RNA or the mRNA or the transcript, and then that is being used to produce the protein through the process of translation that occurs in the ribosome. So what is the phenotype of the cell? Physical characteristics of the cell, right? And the physical characteristics are determined by the activity of the proteins that are being produced, right? We talked about that. So you have these proteins that are needed, that are generated in the cell, they provide the specific characteristics that the cell has in its environment, okay? As far as the characteristics of RNA in bacteria, you should remember that there are 3 types, right? We talked about that. They include messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA or rRNA. You should remember that the RNA, for the most part, is going to be single-stranded in bacteria as well as other cells. And then we also have a base switch out between thymine is going to be switched for uracil in RNA. So everywhere that you would expect to see a T, you're going to see an U, right? So that, and remember that in relation to this, because there's going to have this kind of thing again, where you have the base pair matching between the DNA, which is this top strand. Does everybody see that? How do you know it's DNA? Because it has what? T's, right? This bottom strand is RNA because it has U's in it, right? So, and remember that, of course, that the process of protein synthesis is occurring in the ribosome. tRNA is, we've talked about, is the one that has the anti-codon sequence, matching up to the codon sequence on the mRNA, and that is what's carrying in the right, the amino acids, for protein synthesis. I'm not gonna have anything like this for you guys to label or match or anything, but it shows you the interrelationship between the anticodon sequence of the tRNA molecule in relation to matching up base pair-wise with the codon sequence on the mRNA strand. And then also at the very top, you can see that the part of tRNA that's away from or opposite to the anticodon has the amino acid attached to it, and that's how amino acids are carried into the ribosome, is on the tRNAs. Okay? Now, as far as transcription is concerned, remember that there is one enzyme that's associated with this process. It's called RNA polymerase. RNA polymerase does not require priming. DNA polymerase does. Okay, let me repeat that. RNA polymerase does not require priming to get started. DNA polymerase does. What enzyme is responsible for priming our DNA polymerase during DNA replication? DNA primase, right? Because it puts down the RNA primers, and then that's what's going to initiate the activity associated with DNA polymerase 3, for example. So RNA polymerase does not require priming. You can have the promoter sequence, as we've talked about, that is associated with the gene that can be basically recognized by these specific subunits of the RNA polymerase known as sigma factors. Sigma factors are going to help to orient the transcriptional process associated with the gene. And this core enzyme, which is part of the RNA polymerase, is going to start reading through only one strand, right? It's not both strands. It's going to read through only one strand of the gene sequence, and that's called the negative strand, negative sense strand of the DNA sequence in the gene. Remember that our mRNA is being elongated or synthesized 5 to 3, and the negative strand in the DNA is being read in which orientation by RNA polymerase 3 to 5. Okay, it's always read 3 to 5. Down here at the very bottom, we always have those 3-step processes. Remember, we said that there is always going to be initiation. What's the second step? Elongation. Okay, and the last one is termination. Okay, so for termination or transcription is associated for this particular aspect is the hairpin loops that's in the mRNA, causes the disassociation of the RNA polymerase from the DNA strand, and then transcription stops. Okay. Again, there's an illustration here showing you the process of transcription, initiation, elongation, and termination, just as a reference there to look at. Okay. So remember that only one strand is going to be used to generate the RNA from in the gene sequence, and that strand is being read in which orientation? 3 to 5. It's always going to be read 3 to 5, right? mRNA is produced 3. There you go. So we ended this talk with talking about translation. Where does translation occur at? It occurs in the ribosome. The ribosome is responsible for taking the mRNA and producing what from it? Protein, right? So in order to do this, the codon sequence associated with the mRNA has to be read through, and then amino acids are going to be represented for each codon sequence that is placed within that mRNA that's being used in the ribosome. Remember that there are 3 bases associated with one codon sequence that encodes for a single amino acid. There are 64 possible combinations, and there are 3 of those that do not encode for an amino acid. There are 61 of them that do. Okay? What are the 3 that do not encode for an amino acid? The stop codons. Stop codons do not encode for amino acids. The rest of the 61 do. Okay? We have 3 stop codons. All right? So again, going through this process for transcription, we talked about transcription. Translation is the same thing, right? Initiation, elongation, termination, right? So what is going to kick off this whole thing? The smaller subunit of the ribosome is going to associate with the mRNA around what is called the ribosomal binding site. This is going to pin up the start codon. Everybody remember the start codon sequence? NADPG, codes for FMET, which is a formal added group to methionine, and this is gonna be carried in by the tmRNA. Remember that the elongation complex is going to form when you have the 50S combining with 30S to produce the fully formed ribosome. And the ribosome is what's responsible for reading through these codons and producing the elongated protein within the cell. So from this slide, it's pretty busy. I don't really care if you guys remember the sites of the ribosome, so we'll just not worry about that. But I do want you guys to remember this as far as the process of elongation. What happens with the ribosome is that as it proceeds down the mRNA, okay, it's moving or translocating one codon at a time. How many bases is that at a time? 3 bases at a time, right? And it's going down the mRNA in this orientation here, which is 3 to 5, right? So it's going, or not 3 to 5, 5 to 3. So it's going 5 to 3 down the mRNA, one codon at a time. How many bases at a time? 3 bases, right? And it's going to keep doing that until it gets to what? Stop codon, right? So there are 3 stop codons. You don't have to remember the sequence of those. Just remember that there are 3 of them, and they do not encode for amino acids. Okay? They only encode for stop. So there are release factors that are going to bind to the sites where the stop codon are located at, initially in the A site, and then this is going to cause the ribosome to fall off and release the protein. Protein is going to go do things for the cell. All right? So everybody got that? So make sure you remember the start part of this whole thing. I think that's the biggest part of this. Remember that the ribosome is going to associate with the mRNA, and it's going to move down the mRNA 5 to 3 prime. Again, producing the new protein by combining the amino acids together. Oh, I forgot about that. So I did ask you guys on your last quiz about what type of bond is formed between the amino acids. What is that called? It's called the peptide bond, right? So peptide bonds are formed between the amino acids that actually generate what is called the primary structure of proteins, which is the amino acid sequence. Okay? So that's it as far as this is concerned. I'm not going to ask you anything on any of that. Any questions about that? Yep.
So when you're saying 30S and 50S, are you talking about the different halves of the ribosome?
Yeah, they're different subunits. They have different molecular weights. So that's what they have with S, which is the biochemical term. So that's all it is. So the smaller one associates and then the bigger one comes in after that.
Do we need to know like the 50S and the 70S?
Just from the standpoint, yes, because the ribosomal binding site on the mRNA is only to be recognized by the 30S subunit, okay, initially. It's not recognized by the 50S subunit, so it's the smaller of the 2 subunits of the ribosome that's going to match up around the ribosomal binding site on the mRNA. So you do have to remember that, okay?
All right.
Any other questions about that? Okay, all right. Oh, and let me ask you this. Does translation occur, transcription occur in the nucleus of bacterial cells? Is that true or false?
False.
Why?
This is not a main protein.
No, remember transcription we talked about is DNA to RNA.
Yeah.
mRNA. So does that occur in the nucleus of the bacterial cell?
No.
Why not?
It's not making RNA.
Because what?
It's not making RNA.
No, it is making RNA. No. So remember, transcription again is dealing with DNA to mRNA, and what the big part of this is, does this occur in the nucleus of the bacterial cell? No, yes. Can I ask you this? Do bacterial cells have nuclei?
No.
So it can't, right? Because it doesn't have a nucleus. Okay? Remember we talked about that, right? Bacteria, we had that on first exam, do not have nuclei. So if they have no nucleus, they can't have transcription occurring there because it doesn't exist, okay? All right, everybody got that? Maybe, I don't know. Okay, so the third part of the genetics talk was looking at mutations and also genetic transfer through what is called horizontal transference in bacteria that included transformation, transduction, and conjugation. So as far as mutations are concerned, we talked about how there is inherited genetic material that can occur through this process, which is dealing with cellular division, binary division. And what is this called? This is called vertical transference of DNA. Then we had the other part of this, which was dealing with two cells that are not going through cellular division, but they're passing genetic material, okay, within the environment, okay, and this involves a donor cell, and what's the other cell? Recipient, okay, and this does not involve the cellular division. This is called what type of transference? Horizontal. Okay, we'll talk about those three mechanisms of horizontal transference in a minute. Okay, so as far as mutations are concerned, spontaneous mutations. Okay, spontaneous mutations can have three different flavors to them. First one is the one I really want you guys to study here on this one, which is base substitution. So remember we were going through the two strands, and I came up with a strand that was like 5, A, T, G, C, A, T, 5. And so, like I was saying, with the DNA polymerase 3 is going to read that template strand in that direction, right? Because it always reads it 3 to 5, right? So when it starts reading through this, it's going to have a 5' in there, and you'll have an A here, T here, G here, A here, C here, A here, T here, and 3' in. Is that correct? Why not? There is a misstep here, right? So that is a base substitution mutation that occurs on the strand because of a lack of proofreading by the DNA polymerase. So it's called base substitution. There are two phenotypic mutations that can occur as a result of a base substitution mutation. You have what's called a missense mutation, which actually generates a protein that has an amino acid change to it, but it's still functional, okay? Then you have what's called a nonsense mutation phenotype, which actually has a stop codon in it. So it truncates the protein is not functional, therefore that protein is not going to be produced in the cell, okay? So make sure you guys study this, base substitution. Remember this, right here, this is a base substitution mutation. You see something like that on the test, okay? I'll highlight that for you to make sure you guys remember that. But there is a base substitution that occurs that can cause a mutation generated in a gene sequence of the DNA strand, resulting in a missense and or a nonsense mutation. The other two that I don't really care if you guys remember any details on this except for the names, okay? So we have the frame shift mutation is the other type of spontaneous mutation that can occur in bacteria. Then the other one is transposable elements or jumping genes, okay, that are associated with generating spontaneous mutations. The one that I'm concerned with, again, is the base substitution mutation that we talked about, because that is logical. You guys should know something about this in relation to DNA sequences, base pair matching, and stuff like that. As far as the horizontal transference mechanisms in bacteria, there are 3 of these. They're all summarized up here: transformation, transduction, conjugation. Remember, transformation involves non-cellular or naked DNA that is free out in the environment, binds to a cell. That competent recipient cell is going to take it in and then use it internally. Transduction involves DNA exchange between the donor to recipient through what? A virus, right? And then the last one is conjugation. Conjugation is known as bacterial sex. This is a direct contact that occurs between the donor cell and the recipient cell, allowing for genetic information to be passed through the sex pilus from the donor cell to the recipient cell, okay? The rest of that, don't worry about down at the very bottom. Just remember those definitions up at the top. Don't worry about this slide. Don't worry about this slide. Don't worry about this slide. Yes, don't worry about this slide. So I do want you guys to know that fertility plasmid transfer in E. coli that involves the F positive and F negative cell, right? So the F positive cell is the donor in this case. The F negative cell is the what? The recipient, right? So the F positive is going to have the sex pilus with the fertility plasmid or F plasmid that's going to be transferred by conjugation through the sex pilus into the F negative cell. And then what happens to the F negative cell as a result of that? It becomes an F positive cell and has a sex pilus on the surface, okay? So there's an illustration that is right on the next slide. I will not have something like this for you guys to label on the test or anything, but it shows you the stepwise process and how that fertility plasmid is transferred from the F positive to the F negative, resulting in the two phenotypes of being F positive in the very end, okay? And that's it for that one. Any questions? Before we move on? You guys okay with that? Don't worry about any specifics about transformation or transduction. The only one I want you to know anything about is conjugation between the fertility plasmid associated with E. coli and the F positive and negative cells. So genetic therapy, we did talk about, genetic therapy being an important part of medical technology and how that's exploded on the market. There are multiple things to remember in relation to the different types of medicines that are available that are genetic therapies. Don't worry about this slide. Definitely for this one, there is the transfer of the genetic information into the kid that was suffering from SCID, all right, and replacing the specific enzyme called adenosine deaminase or ADA. Remember this, okay, since this is microbiology, that this uses a viral vector to deliver a correct copy, a functional copy of the ADA gene into the patient's cells. Okay, this was a positive result because this individual recovered to live a normal life after that occurred. There was another story out there that had a negative endpoint to it, which was associated with the transfer of the gene associated with transmetabolase and ornithine transmetabolase deficiency or OTC. And this one utilized a specific enzyme or a specific vector, adenovirus vector, to deliver the correct copy of the OTC gene into the patient. And then what happened to the patient? They died from it. Okay, so an adverse event associated with the transfer of that material through the adenovirus that led to septic shock, and the person died from it. So two different outcomes, two different stories there. Remember that viruses were involved in transferring this information, genetic information, into the cells to deliver the correct copy of the gene so that they can produce the gene and produce protein from the gene as a result of that transfer. Don't worry about anything on this. Don't worry about anything on this. All right, so as far as the CRISPR-Cas9 system, so everybody should know this because this is a hot commodity. Very expensive to use, and it's being used for T-cell therapeutics and other things as well, also sickle cell. So make sure with CRISPR-Cas9 that this is associated with the CRISPR component, and so there are parts of the DNA fragments that are derived from a bacterial phage infection here. You don't have to remember what the cluster regularly interspersed, you don't have to remember any of that. Just remember CRISPR-Cas9 is actually the protein that's involved in aligning the RNA-guided DNA recognition endonuclease and removes foreign DNA from the cell. So you've got the CRISPR and the Cas9 that are going to be working together to and replacing it with the correct copy, right? And, so as far as this kind of goes through, again, a little bit of a summary there. I'm not gonna have any illustration on the test about this. But so the whole idea with this particular system that's different from the other two that we talked about with the viruses, those viruses are just delivering a correct copy of the gene into the cell, but they're not taking out the mutated copy, all right? So the CRISPR-Cas9 can actually take out the mutated copy and then replace it with the correct copy of the gene, okay? So that's one of the things that's important. So this two steps right here, removal of mutated sequence, and insertion of the correct gene, so it can do two of those things, right? So this one we talked about, and since this is microbiology dealing with HIV, which is a viral infection, remember this one involves the CRISPR-Cas9 system that's taking out a functional gene that produces a correct protein and replacing it with a mutated gene, creating a mutated protein in the CCR5 protein receptor, and this makes the patient or the individual resistant to HIV infection because it cannot recognize the cell if it has a mutated CCR5 gene, okay? So in this particular case, this is happening in a clinical trial right now with an individual that has this done to his T cells. They take his T cells, they put the CRISPR-Cas9 technology in there, replacing the correct copy, okay, emphasizing the correct copy of his gene with a mutated form of the CCR5. Why is that necessary? Because a mutated form is resistant to HIV, okay? That's the important part. The mutated form of the CCR5 gene receptor is resistant to HIV infection. HIV can't recognize a cell because of that mutation, right? Has everybody got that? So it's going backwards, right? So we've been talking about replacing mutated genes. This one is doing the opposite, but you can still do it with CRISPR-Cas9. In this case, they're taking out the correct copy of the gene and replacing it with a mutated copy so the virus can't get in the cell, okay? So in this particular case, in this clinical trial, they're using this to, again, take out the gene copy that produces the correct protein, the CCR5 receptor on the outside of the T cells, and placing a mutated copy of the gene in there so that the virus cannot recognize the cell any longer. And if it can't get in the cell, it can't replicate, so HIV is going to be eliminated from the T cell population. Does that make sense? Okay. Of course it does. You could have thought of that, yeah. So, any questions about this, okay? Viruses for the first two, the adenovirus and viral vectors associated with OTC, AD8 genes that are just injected into the cell, replacing the mutated, not replacing it, but basically expressing over the mutated form. CRISPR-Cas9 doesn't do that. What does it do? It takes out the mutated gene and puts the correct copy in, or it takes out the correct copy and can put the mutated copy in, okay, as in the case with this clinical trial, okay? So, any other questions? Okay, any other questions? All right. Last one, looking at antibiotics. So we talked about this timeline, but I'm not going to have any questions on this, but just remember the golden age of antibiotic discovery was between like 1940, 1970, 1941, and 1968 specifically, in that time frame. So about 30 years or so. So the other part of this was looking at penicillin. Make sure you know who discovered penicillin in his laboratory, which is Alexander Fleming. He produced it on, or recognized it on a plate of staph that was being inhibited by the penicillium fungus that was growing on it, and he was trying to isolate this out. And this was represented here, where you have the penicillium nonium down here at the very bottom that's inhibiting that staph that's growing on the plate. You can see that little zone right there. So he couldn't do it, and there are two guys out of Oxford University that picked this up. These are chemists, Howard Corey and Ernest Chain. Both of these were the chemists that actually worked to purify penicillin for human use. And so the first patient that was dosed, make sure you know his name. The first patient that was dosed was Albert Alexander with penicillin. Did he live? He did not. He did not live. And does anybody remember what he had? What was wrong with him? He was working where his rose bushes scratched his eye, right? And they had to remove his eye because he had a bad infection. But you don't have to remember that because that's just making sure you guys are listening to me on that. But that's pretty much it from this. So remember, Howard Corey and Ernest Chain were the two chemists out of Oxford that actually successfully worked on the purification technique that was actually again approved for human use and started dosing it then. Okay. So we did talk about how the timeline for antibiotics has been modified a little bit in the last 50 years or so. And so we see that a lot of things are not being produced in a new form. There are changes that have occurred in the last 10 years or so, refocusing on gram negatives. None of this is going to be on the test or anything, but the golden age of antibiotic discovery preceded this with the discovery of a lot of the compounds that are still in use. So make sure you study the different classes of antibiotics that are listed here. So the ones that I want you guys to remember for the test, as far as remembering what class they belong to, as far as their names are concerned. Okay, everybody ready? Okay, so cell wall synthesis inhibitors, make sure you remember penicillin and vancomycin belong to that class. Okay. Protein synthesis inhibitor, make sure you remember tetracycline belongs to that class. DNA replication inhibitor, make sure you remember ciprofloxacin belongs to that class. RNA synthesis inhibitor, rifampin, belongs to that class. Tetrahydrofolic acid synthesis inhibitors, only two of those, trimethoprim and sulfonamide, that belong to that class. They're a combination drug. For the cytoplasmic membrane damager, I definitely want you guys to remember the name colistin. So colistin is a big name for gram-negative treatment clinically. And then the rest of it, you have to remember any names or anything like that. So those are the ones I want you to focus on, and remembering what class each of those belong to. So fatty acid, but also the mechanisms of these things. Everybody should know fatty acid synthesis inhibitors are involved in inhibiting fatty acid synthesis in bacteria, stuff like that. Yeah.
What was the last one?
Which one? Right there, where it says polymyxins in parentheses, it says colistin. Okay, so colistin, remember that name is a cytoplasmic membrane damager. Okay, and it's used to treat what type of infections? Gram-negative resistant infections, right? DR infections. All right. Any questions about that? I think this is on your homework, right? Is it? Has anybody done their? No. All right. So nothing to illustrate here. I'm not going to ask anything about resistance here. As far as defining resistance, you should know what resistance means, that the antibiotic can no longer inhibit the growth of or kill the bacteria. Okay, so it's lost its effect. Okay. As far as mechanisms and how this occurs, there are these two slides down at the bottom. So you have the drug inactivating enzymes, modification of target molecule pathway. We talked about these two examples, penicillinase, which is an enzyme that breaks down what is the substrate, penicillin, and penicillin can't get in the cell, so it can't inhibit the cell walls that are being formed. Then we have the modification of the PBP, which was the penicillin binding protein. Does anybody remember why this is pertinent for penicillin? Because this is how staph developed resistance to penicillin, is it had PBP mutations where penicillin could no longer recognize it because the target molecule was modified, which was the PBP protein. Then we have the other components associated with the ability to become resistant, which is increasing or decreasing the influx or uptake of the antibiotic, so it blocks it from getting in the cell. Gram negatives do this a lot with their outer membrane. Increased elimination of the internalized drugs, so what they do is they pump this back out into the extracellular area so the antibiotic can't remain in the cell to elicit a response, okay? Then on the next slide, this shows you the illustrations of those 4 types of mechanisms of mutations or resistance phenotypes that can occur, mechanisms of resistance, get my words right here in a minute, for each of the 4 types that we just talked about. It shows you an illustration of how this works. First one is decreased penetration into the cell, so it looks like it's just bouncing off the membrane. Production of drug inactivating enzymes, so remember penicillinase breaks down what? Penicillin, right? So it breaks down penicillin, rendering it ineffective. Number 3, modification to the target. So that means that, for example, you see that one there, you can no longer bind to the targets because it's mutated, and that is associated, we said as an example of PBP, with penicillin and staph, right? Okay. You also have the pumping associated with the increased elimination of the drug that's been internalized in the cell. If you look down here, you can see it goes in the cell, it's pumped back out through these efflux pumps, okay? So that is what you need to remember as far as the mechanisms of resistance are concerned, okay? Nothing is going to be on this slide. Okay, so there was the AMR report, or the Antibiotic Resistance Threats Report, that was put out in 2019 by the Centers for Disease Control, and this basically was going to look at different statistics associated with drug-resistant infections. And the main thing that I wanted you guys to remember from this was the 3 different classifications that the CDC placed on different organisms that are associated with either being urgent hazard level, serious hazard level, or concerning hazard level. Urgent hazard level is the highest threat to public health, right? So, the names I want you to remember that are associated with the urgent hazard level threats include C. difficile, carbapenem resistant, Acinetobacter and Enterobacteriaceae, and then drug resistant Neisseria gonorrhea. Well, except for yeast, I'm not going to worry about Candida auris, okay, because that's a yeast, that's not a bacteria, but still. So those are the 3 that I want you to remember for that. That would be C. diff, the carbapenem resistance, and then the drug resistant Neisseria gonorrhea. Don't have to remember any of the other examples for each of the other 2 classes. Just remember that those 3 organisms or types of organisms exhibiting certain types of resistance are associated with the urgent hazard level threat class associated with the antibiotic resistance report by the CDC. Okay, so don't remember any of the other names on there.
I have a question.
Yep.
We're supposed to know, according to parentheses, for the carbapenem resistance?
No, I mean, you don't have to.
Okay.
Because the Enterobacteriaceae is a family that has Klebsiella and E. coli in it, so.
Okay, so just the family then.
So family is more important because you get multiple types that are carbapenem resistant clinically. So could be E. coli, Klebsiella, Proteus, all of those belong to the same family. And nothing on this, nothing on that. KPC, we talked about this as a carbapenem resistance mechanism known as Klebsiella pneumonia carbapenemase, or KPC. This is a transmittable resistance mechanism. So what does that mean? Goes back to this, right? Horizontal transference, right? These cells can pick up this KPC, from just genetic information being passed back and forth between different gram-negative organisms. So we do this a lot, and this generates resistance to carbapenems, which are used for treating gram-negative infections. The rest of the stuff that's up here, don't worry about it, okay? So just remember KPC associated as being a transmittable resistance mechanism form that is associated with gram-negatives becoming carbapenem resistant, okay? Any questions? Any questions? Said details, any of the details, if I said you need to know enzymes or anything like that, focus on that, okay? So it's gonna require some memorization, you may have to go back and